Hello. Good afternoon.
Hello.
Hi, everybody. Come on in. Type your name in the chat for me if you would. Thank you for coming this afternoon. It's good to see everybody. All right. Yes, if you would type your first name or your nickname into the chat for me. That's my record that you've been here today. All right. Good to see everybody. Okay, so some of you may have seen an announcement that I sent out about a half an hour ago. I'll just take a minute to explain that if you didn't see it. One of your classmates actually contacted me about this week's lecture, which is about immunology. And if you haven't had a chance to look on YouTube at that lecture yet, it's a long one. There are 6 individual videos for that lecture. It's a long one. And if you go to our syllabus on Canvas and you scroll down to the tentative schedule at the bottom, immunology is actually scheduled on 2 days. In other words, if we were together and I was lecturing to you in person, I would have taken 2 days to finish that immunology material. So that means you shouldn't be expected to finish that lecture material till sometime on Monday next week, which means the quiz should not be due until Tuesday at midnight. So I originally had the quiz marked as being due tomorrow by midnight. I have now extended that to Tuesday. So there is no lecture quiz due tomorrow. The immunology lecture quiz will be due Tuesday at midnight. Okay, and then you'll be moving on after that to vaccination in lecture. So we are in the final part of the course in terms of lecture material. We've now left the cellular mechanisms of microbial life. We've left metabolism and we've left genetics. And we're moving on to the last part of the course, which is really much more about how microbes impact public health. I'll say medicine and public health. So the final several topics include things like immunology again, an introduction to immunology, an introduction to vaccination, an introduction to epidemiology, and then the final one is an introduction to the pathogenesis of disease, how microbes behave as pathogens and cause these infectious diseases. So I'm hoping that you'll enjoy these last topics because a lot of you I know are interested in careers in healthcare. And so I think that, and coupled with the fact that we're living in a pandemic right now, I think these last several lecture topics are very applicable to what's going on in the world right now. Now, in laboratory, remember, we're just a little bit behind. So while we've moved on in lecture, we still have a little bit more work to do in the laboratory. Today, we're going to be talking about the last biochemical test that we're going to talk about, which is having to do with respiration, particularly anaerobic respiration. And that will finish us up in terms of learning about culture media and testing that we do in the microbiology lab. All right, so I'm going to go ahead and pull up our schedule on Canvas just so we can all remind ourselves of everything for a minute. Let me put this page down first. Okay, so I'm just up in the modules section. Here we are, we're in week 12, the week of April 5th. You had the exam on April 5th, and then you have this lecture topic about immunology. And you could see the quiz, the date for the quiz has been bumped to next week. We did talk about protein catabolism when we were together on Monday, and today we're going to be talking about respiration. Now remember, because our schedule got bumped and jostled a little bit, your laboratory questions this week are not about respiration, but they're about protein catabolism. Okay. Next week, your final lab questions will be about respiration. Okay. So this week, even though we're talking about respiration today in lab, your lab homework is about that protein topic, protein catabolism. And your last lab questions will be due next week at the end of the week. Now, just because we will be finishing up in terms of learning about laboratory testing, we're not quite finished in laboratory because we're going to have an assignment to sort of wrap things up, which is identification of an unknown microbe. So looking ahead to next week, which is the week of the 12th, during lab on Monday, we're going to talk about this sort of final assignment, this sort of wrap-up assignment, where you're going to be looking at a set of data and you're going to be analyzing that data in order to identify an unknown microbe. So you can see that there's actually a link here on Canvas where all of the materials can be found for this assignment. Again, we're going to talk about this. We're going to spend our time in lab on Monday talking about this. But you can see there's an instruction sheet. There's a couple of different data sets. And then there are some keys, dichotomous keys that we're going to be talking about how to use and so on. And once we get, after Monday, once we've talked about this assignment, identifying an unknown microbe, once we've talked about it, you're going to have a week to go through the data and make your identification. I will also, so you can see this is where you're going to be submitting the assignment once it's all finished. And you can see the due date on it is April 19th. Now, on Wednesday next week, you have your last lab practical exam. It's hard to believe that, but we're getting close to the end of the semester. And of course, this lab practical will cover everything that we've done in lab since lab practical exam 2. So this is the link for that. You'll be taking that during the day next Wednesday. As always, sometime between 8 a.m. and 8 p.m. You just need to get it to me by 8 p.m. And then here's that lab homework about respiration. Your last set of homework questions about respiration. After that, I don't have anything posted yet for you, but as you can see, we're rapidly coming to the end of the semester. There's a couple more weeks after that we'll be covering the last lecture topics. We'll be talking in lab about the SARS virus, about the coronavirus and COVID-19. And then the ultimate, the last week of class, which is final exam week. So today we're going to be talking about respiration. So does anybody have any questions about the schedule? Does everybody understand that I've pushed the immunology lecture quiz ahead a little bit? I want to give you plenty of time to watch those videos and take a good set of notes for yourself before you take that quiz. Immunology is a massive subject. I mean, you can take an entire course on immunology, right? Some courses about immunology last 2 semesters. So, what we're doing is just a very quick sort of overview of immunology in this course. But it's important because we're also talking about, we're talking about vaccination and we're talking about infectious disease. So it's very, very helpful for us to have some understanding of how the human immune system works. All right. All right, so if there aren't any questions, what I'm going to do is go ahead and start into the lab exercise. As always, if a question comes to you, just type it into chat and we will talk about it. Okay. All right. So we've been talking about metabolism now for a couple of weeks. We've talked about how carbohydrates are metabolized, specifically catabolized, taken apart through both cellular respiration and through fermentation. We've talked about tests. We talked about the OF test, which determines whether a microbe is oxidative or fermentative or both. We've talked about the carbohydrate fermentation test, which allows us to test for the ability to ferment different sugars. We talked about tests that look for an organism's ability to break apart protein. We've talked about tests that examine whether a microbe can take an amino acid apart. Lots and lots of individual tests about metabolism, about the kinds of things, the kind of reactions that go on inside of our microbial friends, the microbial cells that live on us and in us and around us in the world. So today we're going to be talking about a couple of tests that are related to respiration. Now, just as a review, when we talk about cellular respiration, we often use this term oxidative. We talk about oxidative metabolism or oxidative catabolism. Remember, that word oxidative has nothing to do with oxygen. Instead, it's referring to the kinds of chemical processes that are involving oxidation reduction. When we talk about this word oxidation, we're talking about an atom or a molecule losing electrons. When we talk about reduction, we're talking about an atom or a molecule gaining electrons. It's very, very common in living things to see these oxidation reduction types of metabolic reactions. They're just very common in living things. Remember that if something is getting oxidized during a metabolic pathway, something else has to be reduced. It's one of those laws of nature. So we've seen lots of different metabolic pathways. We know that microbes are incredibly diverse. Some microbes are able to do this oxidative type of metabolism. They're able to do cellular respiration. Sometimes they do it aerobically. Sometimes they do it anaerobically. Some organisms are able to do both of those. And remember, when we talk about cellular respiration, what we're really talking about is a long series of reactions that takes the glucose molecule and breaks it down, breaks it all the way down until there's nothing left but carbon dioxide. Glucose gets oxidized in that reaction, broken apart, in other words. And then remember, something else has to get reduced at the end. If you're an aerobic microbe, oxygen is going to be the final electron acceptor. If you're an anaerobic microbe, you're going to use a different molecule. You're going to use nitrate or sulfate or carbonate or any of several other final electron acceptor molecules. And those final electron acceptors are located at the end of the electron transport chain. All of the electrons that are released from glucose, as glucose gets oxidized or broken apart, all of those electrons are going to be sent down the electron transport chain. And their energy is going to be harvested. At the end of the chain.
Question.
Yes. Yes, go ahead. Go ahead, anybody. I can't tell which one of you was called out. Any question? Okay. So.
Yes, I'm sorry. I
Oh,
took
that's
off
okay.
the mute.
That's all
It's
right.
Vicky. No, I just looking at this. Reduction means they gain an electron, and oxidation is loss of an. That's back.
That's right. I'm sorry?
I said that's backwards.
No.
If it's in reduction, it should. I know, but it should be reduction. They lose. That's why I'm confused.
Remember in lecture we said that it's very helpful to use a little memory device here. So I like Leo goes Gur.
Leo, yeah.
Loss of electron, oxidation. Gain of
All right,
electron.
that's what that. Okay,
reduction.
I gotta.
Listen, chemists have those little memory devices because it's hard sometimes to remember which is which. Oxidation, or losing electrons, is another word for catabolism, really. If you oxidize something, like glucose, you're breaking it apart.
Down, got it.
And that's releasing the electrons away from the glucose. So the glucose is getting oxidized. Okay, I know the language is weird. It just,
It's backwards.
Seems strange, right?
All right. Thank you.
You're welcome. So lots of microbes do cellular respiration, and then the follow-up reaction, which is oxidative phosphorylation. Again, oxidative because this is an oxidation reduction reaction. And phosphorylation because we're going to take phosphate groups and put them onto ADP to make ATP. This is where the proton gradient comes in that we talk about in lecture. All of the energy that got harvested from those glucose electrons, all of the energy that was pulled out of them in the electron transport chain is going to be used to do some work. And that work is pumping hydrogen ions out of the cell so that we build a concentration difference or a gradient. That gradient is then going to be allowed to resolve or go away or equilibrate. The hydrogen ions that get pumped out of the cell are then going to flow back into the cell through that very special molecule called ATP synthase. Remember, this is a molecule that sits in the membrane of a microbial cell. After all those hydrogens get pumped out of the cell, they're going to flow right back in through a channel, through a tunnel in the ATP synthase molecule. And as they flow back in, as they physically flow back into the cell, they are literally going to spin part of that molecule, part of that ATP synthase. And it's the spinning motion that puts the phosphate onto the ADP and creates ATP. ATP synthase is a molecule that shows up in all living cells. That tells you that this process of oxidative phosphorylation must be very good, very efficient, because all living things use it to make ATP. Now, fermentation is another pathway to make ATP, isn't it? But fermentation doesn't involve oxidative phosphorylation. Instead, it involves substrate level phosphorylation. And remember, this kind of adding a phosphate group to make ATP, this kind involves an enzyme, and the production of ATP is just piggybacked onto the reaction that that enzyme normally performs. The enzyme is doing some reaction. It's helping some substrate to become a product. And along the way, ADP gets phosphorylated to become ATP. Oxidative phosphorylation is very efficient. In other words, for a small amount of glucose, you're going to get lots of ATP made. Substrate level phosphorylation is very inefficient. You get 1 ATP every time the enzyme catalyzes its reaction. So very efficient, very inefficient. And yet, fermentation is very common in the microbial world. There are some microbes that only do fermentation. And that's because they're microbes. They're not big, complex, multicellular organisms like you and I. They can survive with the small amount of ATP that gets produced through fermentation. Lots of microbes can do more than one of these pathways. And we use metabolic ability in order to help us identify microbes, because each microbe is a little different than the next and very, very diverse. All right. So I'm going to get into a little bit of nitty gritty here about what's happening at the bottom of the electron transport chain, because that's what we're going to be able to test for with today's laboratory test. In an aerobic organism that's doing cellular respiration, remember, they're using oxygen at the end of their electron transport chain. So as the electrons pass down the electron transport chain, their energy is being harvested from them. And when they get to the bottom of the chain, they're going to be accepted by oxygen. So the oxygen molecule, because it's going to gain these electrons, is being reduced. We're at the bottom of the electron transport chain here. We're at the end of cellular respiration. Oxygen plus those electrons that came out of the glucose, plus a couple of hydrogen ions, I forgot to add those in, plus some hydrogen ions, are going to create water. Remember, long at the beginning of the path was a glucose molecule. The glucose got oxidized, something else got reduced. Then that thing got oxidized, then that something else got reduced. Then the next thing oxidized, then the next thing reduced. And it just keeps happening all the way through glycolysis and pyruvate processing and Krebs cycle. It's oxidation reduction, oxidation reduction, oxidation reduction. So at the very end, there has to be a reduction, because every oxidation has to be matched with a reduction. So at the very end, oxygen acts as the final electron acceptor. It's going to take all those electrons that came out of the glucose, along with some hydrogen ions, and it's going to make water. This is what we can call the pathway to reduction at the end of the electron transport chain. Now, here's some information we haven't really talked about yet, except very briefly in that lecture. In the electron transport chain, what we're really looking at are these big groups of proteins that are capable of harvesting energy out of electrons. So the electrons come into this chain of proteins, and they have to be passed from one protein to the next, to the next, to the next. There are a couple of different molecules that do that passing. One is this molecule called ubiquinone, which we talk about in lecture. And the other one is a group of molecules called cytochromes. So these are molecules that are very easy to both oxidize and reduce. In other words, they can very easily accept an electron from a protein and then pass it on to the next protein. cytochromes act like the middlemen between the protein complexes that make up the electron transport chain. They help get the electrons all the way down through the chain. Now, it turns out that there are 4 different families of these cytochromes, at least in bacteria. There's type A, type B, type C, and type D. So different species of bacteria use different cytochromes in their electron transport chain. Now I've got cytochrome C highlighted here because we can do a test in the laboratory related to cytochrome C. We'll talk about that test in just a minute. But first, let's look at what the anaerobes are doing. Remember, aerobes use oxygen as their final electron acceptor. Anaerobes use a different molecule. But other than that, they're still doing the same cellular respiration process. They're taking glucose and they're breaking it apart until there's nothing left but carbon dioxide and a bunch of electrons. Those electrons are going to get fed down through the electron transport chain, through that chain of proteins, with the cytochromes helping to pass the electrons from one protein to the next. At the bottom of the electron transport chain, there still has to be a final electron acceptor. It's just not oxygen. Most of the time, it's either nitrate, sulfate, or carbonate, with nitrate being the most common. These 3 molecules, nitrate, sulfate, and carbonate, have oxygen in them, but they don't have any carbon. They are inorganic. Most of the anaerobic organisms that do cellular respiration use one of these 3 as their final electron acceptor, with nitrate being the most common. There are some microbes, though, that use other final electron acceptors, including molecules like fumarate, triethylamine, and even a molecule called DMSO. We haven't talked about these chemicals because these are not commonly encountered as final electron acceptors. These are organic molecules. These are carbon-containing molecules, and they can accept the electrons. They can also be reduced at the end of the electron transport chain. But this is an uncommon set of chemicals. So we're going to focus on nitrate in the laboratory because it is so commonly used as a final electron acceptor. We looked at the pathway of reduction for oxygen. Now let's look at the pathway of reduction for nitrate. That's what I have in green here. Nitrate, which we've talked about over and over, but we haven't really looked at, is a combination of nitrogen and oxygen. When nitrate accepts electrons, when it gains electrons at the bottom of the electron transport chain, it's going to get reduced just like oxygen does. But instead of forming water, it's going to form a molecule called nitrite, in addition to some water. Nitrite and nitrate are 2 different molecules. Easy to confuse. Nitrate is what we start with. It's going to get reduced. It's going to gain electrons, and it's going to become nitrite and a little bit of water. Now, for some microbes, that's the end. That's the end. The electrons got accepted by the nitrate, and we reduced the nitrate into nitrite. But there are other other microbes that will reduce even further. In other words, they can also reduce the nitrite. And that's what you're seeing in this second equation. So for some bacteria, they're going to reduce the nitrate to nitrite right here, and then they're going to be done. But some microbes will then take that nitrite and they will reduce it further. They're going to add even more electrons to it. It's going to gain even more electrons. And when the nitrite gets reduced, it becomes a molecule called nitrous oxide. For some microbes, that's the end. They're all done. But for other microbes, they can go even further. They can take this nitrous oxide and they can reduce it even further. So the nitrous oxide can then accept electrons. And it's going to become nitrous gas, or nitrogen gas, I should say, not nitrous. Nitrogen gas, or N2. So we don't see that with oxygen. The reduction of oxygen is pretty simple, chemically speaking. The oxygen molecules accept the electrons, and we get water formed. But in anaerobic organisms that do cellular respiration with nitrate as the final electron acceptor, there are actually different lengths that the reduction will occur. Sometimes the nitrate goes to nitrite and we're done. Sometimes the nitrite is further reduced to nitrous oxide and then we're done. And in other times, the nitrous oxide is even further reduced and we get nitrogen gas. So very, very complex pathway of reduction for nitrate. I also included this reaction and this reaction more just for completion's sake. This molecule is sulfate. It's going to accept electrons and become hydrogen sulfide and a little bit of water. And this molecule right here, this is carbonate. And when it gets reduced, it's going to create methane and a little bit of water. None of these equations here are balanced. Don't worry about that. Just understand that for these very common final electron acceptor molecules, when they get reduced, we know what product is made at the end. Okay. All right. Any questions about what I've said? When I said microbes are diverse, I meant it. Microbes that use nitrate as their final electron acceptor, different species are able to reduce that nitrate to different products. Some of them will reduce the nitrate to nitrite. Some will take the nitrate all the way to nitrous oxide. And some will take the nitrate all the way to nitrogen gas. It's just different pathways, different lengths of reduction pathways. So it's important that since nitrate is such a common final electron acceptor, it's important that we have a way to test microbes so we know which reduction pathway they're using for the nitrate. And that's one of the tests we're going to look at today. Now, we're also going to look at a test called the Oxidase test. Now, the Oxidase test is nice because like the Catalase test, if you can remember that one, this is a quick and easy test. Oxidase test relates to that molecule called Cytochrome C that I mentioned a couple minutes ago. Remember, we said not all bacteria use cytochrome C in their electron transport chain, but some species do. If you have an electron transport chain and you're using cellular respiration as your way to make ATP, you've got some kind of cytochrome in that electron transport chain. It just depends on what type of cytochrome you're using. That varies by species. So there are some bacteria, some microbes that use cytochrome C specifically. In the oxidase test, we are testing for the presence of an enzyme called cytochrome C oxidase, hence the name of the test. Remember what cytochrome C has to do. Cytochrome C is the middleman between all the proteins that are sitting in the electron transport chain. Cytochrome C is going to gain electrons from one protein, that's reduction, and is then going to hand those electrons off to the next protein, that's oxidation. This enzyme, cytochrome C oxidase, is going to allow for that oxidation to occur to cytochrome C. Let me say that again, because that's important. Remember, cellular respiration is just a long, long series of oxidation reduction reactions. Oxidation reduction, oxidation reduction, oxidation reduction, oxidation reduction, all the way down the line. When electrons get to the electron transport chain, it's still happening. They're getting oxidized, something's getting oxidized, something's getting reduced, something's getting oxidized, something's getting reduced, all the way down the chain. It's proteins that are harvesting energy out of these electrons. But there are several proteins in the chain. So in order to get the electrons from one protein to the next, to the next, to the next, all the way down the chain, we need a cytochrome. And in some microbes, it's a cytochrome C. So imagine that this protein, this first protein in the chain, just received a bunch of electrons that came out of glucose. So it gained electrons, so that protein has now been reduced. It's going to harvest the energy out of those electrons. And then it's going to hand them off to the next protein. But it can't hand them off directly. So instead, what it's going to do is hand them to cytochrome C. And then cytochrome C will hand them to the next protein. When cytochrome C accepts electrons, gains electrons, it gets reduced. When cytochrome C gives away electrons or loses electrons, it gets oxidized. In order for it to get oxidized, it needs an enzyme. It needs an enzyme called cytochrome C oxidase. And that's what we can test for. So if the oxidase test comes back positive, we can interpret that as meaning that cytochrome C oxidase is present in that microbe. And what that means to us is that that microbe has the cytochrome C family in its electron transport chain. If a molecule or a microbe comes up oxidase negative, that tells us there is no cytochrome C oxidase in that cell. They could still, though, have a cytochrome. It's just not cytochrome C. It might be cytochrome A or cytochrome B or cytochrome D. Or maybe that's a microbe that doesn't do respiration. It doesn't even have an electron transport chain, or at least it doesn't use an electron transport chain. Okay, so it's a very particular, specific test. It's testing for the presence of cytochrome C oxidase. Now, I said it was a pretty easy test to conduct. Let's look at that. So what we do for this test is we grow up our organism on agar. It's really best done with agar. We don't want to use a broth here unless we really have to for some reason. And what we're going to do is we're going to take our agar plate out of the incubator, and we're going to use a sterile loop to pick up some colonies. We're going to pick up colonies off the agar plate so that we literally have thousands and thousands of cells on the end of our loop. Then we're going to expose those cells to something called oxidase reagent. Again, this is a chemical, this is commercially available. And all we have to do is drip this reagent chemical onto the colony material, onto the cells, and look for a color change. That oxidase reagent, in the presence of cytochrome C oxidase, is going to turn color. It creates a molecule called indophenol blue. Don't worry about that too much. Just know that it will turn this purpley blue color if it's positive. Cytochrome C oxidase is found in cells that use oxygen as their final electron acceptor. They use cytochrome C and cytochrome C oxidase. And we also know they use oxygen at the end of their electron transport chain. The other cytochromes, A, B, and D, are used by microbes that use other final electron acceptor molecules. Okay, so a positive oxidase test tells us something. It tells us there's cytochrome C oxidase present. So that must mean cytochrome C is present. And that must mean that this is an organism that uses oxygen as its final electron acceptor. 3 good pieces of information from one test. So you can use a loop, but if you use a loop to pick up your colonies, you got to use a plastic loop. Make a note of that. It turns out that this oxidase test is a little bit sensitive. So just like the catalase test, if you remember, you don't want to use a wire loop when you're doing this test. In fact, most people just use a sterile swab. Just skip the loops altogether and go to a sterile cotton swab. You can use the swab to scoop up some colonies off of the agar plate, and then you just drip the oxidase reagent on top of the cells and you look for the color change. I'm going to skip this for just a sec so we can look at this. Here we are. So what you're looking at in this image on the upper left, this is an oxidase reaction. You can see that they've used, again, sterile cotton swabs just to pick up some colony material off of a plate like this one down at the bottom. You don't have to pick up any certain number of colonies. You just want to get a bunch of cells on the end of your swab. And then you take this little squirt bottle of oxidase reagent and you just drip it on top. And this is what you're looking for. It happens immediately. And the bluish purple color is very noticeable. It's dark. So this would be a positive reaction. And this would be a negative, no color change. You read these immediately, just like you would with a catalase test. Now, down here at the bottom, again, this is just an agar plate that has colonies growing on it. You can see that this person skipped a step, didn't they? They didn't bother picking up colonies with a cotton swab. They just dripped the reagent right on top of the colonies. You can do that. These are positive because I have a color change. It turned blue, it turned purple. Most people don't do this because if I've just gone to the trouble to make this nice agar dish, I'm probably going to want to use it for some other tests as well. And I don't want to spoil this whole section of the plate by putting oxidase reagent on it. So most people won't do that. Most people will use a cotton swab and will pick up a few of these colonies to do the test. Now, I want you to jot down a couple other things in your notes, a couple of practical things about the oxidase test. The little bottle of reagent that you buy for the oxidase test is relatively expensive. And unfortunately, once it's opened, once it has, the seal has been broken and you open it for the first time, the clock is ticking on how long that reagent lasts. If you work in a really busy microbiology lab, you're going to buy oxidase reagent in, again, a little squirt bottle, like a bottle the size that you might get with eye drops in them. And because you're in a busy lab, you're going to finish that bottle of oxidase reagent within a week or two. So there's no worries about it going bad. But once you've opened it, the clock is ticking and the reagent will not last beyond a week or two. So if you're working in a lab that doesn't do a lot of oxidase tests, there's another way to do this test without having to throw out unused oxidase reagent at the end of a week or two. What you're looking at over here is that other option. It is possible to buy these test strips that have oxidase reagent already in them. So these are just little strips of paper that have basically been soaked in oxidase reagent. You buy these in little sterile packages, so you only use them as you need them. You open up a test strip, you pick up some colony material with your sterile swab, and you just smear it onto the test strip, which this person has done, and you look for the color change. If the test strip turns blue-purple, then you know you're positive. If it doesn't, you know you're negative. So the nice thing about buying the test strips is there's no waste. If I buy a 50 ml bottle of oxidase reagent, as soon as I've opened it, I only have a couple weeks to use it all, or else I have to throw it away. But if I buy the test strips, each strip comes in its own little package, little sterile paper package. So I open it and then I use it. So those last a lot longer in the laboratory than a bottle of reagent would last. Does that make sense? So you can see both things. You can go into laboratories where the oxidase reagent is in a squeeze bottle, and you can go into laboratories where the oxidase reagent is on a paper strip. It's the same test. It's the same test. You're still looking for a blue color change to tell you that this microbe has cytochrome C oxidase in it. Okay. So now let's talk about this next test. This test is one of the most complex tests that we've talked about in this course, which is kind of why we save it for last. Remember that we said that microbes that do anaerobic cellular respiration oftentimes use nitrate as their final electron acceptor. The nitrate is going to accept or gain electrons and become reduced. Some microbes will reduce it to nitrite, some to nitrous oxide, some to nitrogen gas. This test is going to tell us. This test is going to tell us what is what regarding nitrate reduction. Okay? Now, this test is testing for the ability of a microbe to reduce nitrate to nitrite. So we're going to look for a color change that tells us that there is nitrite in the tube. This test uses basic nutrient broth, basic nutrient broth that has been supplemented with nitrate. You add in a whole bunch of nitrate. Usually it's in the form of potassium nitrate. That's not really important. It's just a little detail here, but we're adding the nitrate in to make it available to the microbe. You're going to inoculate your test nitrate reduction test tube. You're going to incubate it. We generally incubate it for 48 hours. Now that's a little bit different than we're used to. We usually see our tests run for about 24. There are a couple of exceptions, but usually we incubate about 24 hours. The reason we go to 48 hours for this test is because we want to make sure that there's been enough time for the cells to grow, to divide and divide and divide, and have enough time to do as long of a reduction reaction as they want to do, as they're able to do. So we give them a full 48 hours for this test before we read it. Now, the next day, or I should say the next 2 days, when you come in after 48 hours of incubation, you're going to take the tube out of the incubator and you're going to add not one reagent to it, but 2. Now, these 2 reagents have specific names, but we're not going to worry about them. We're going to call them reagent A and reagent B, because that's what most people call them. You'll also hear them called reagent 1 and reagent 2, because that's, again, an easy way to think about 2 different reagents. You put both of them in. They're liquid. You just drip them in to this nutrient broth tube. You're going to check at that point for a red color. The broth starts off as sort of a beige color, very boring looking beige color. You're going to put the 2 reagents in and look to see if it turns red. If it turns red, it means there is nitrite in the tube. And if it turns red, we know that those cells must have reduced the nitrate that we put in the tube to nitrate. Pretty straightforward. Here's where it gets complicated. If you put the 2 reagents in and there's no color change, you're not done yet. You have to do some more work before you know if you have a positive or a negative. If I put the 2 reagents in and it turns red right away, that's a positive result. The organism has reduced nitrate to nitrite. There is nitrite in the tube, and the tube turns red. Positive result. If there's no red color, after I add the 2 reagents, I'm going to add a 3rd chemical into that tube. And that 3rd chemical is just a zinc reagent. It's basically zinc metal. Now, why would I do that? Well, it turns out that zinc is also able to reduce nitrate to nitrite. So when I put the zinc in, if there's any nitrate in that tube, it's going to get reduced to nitrite, and the tube is going to turn red. But here's the kicker. That means the microbe didn't make the nitrite, the zinc did. And that is a negative result. Let me say it again because it's very important. That broth that we made up in the lab, that nutrient broth, we added a bunch of nitrate to it. And then we put some cells in and we let the cells grow. Now, if those cells were able to reduce that nitrate into nitrite, I'm going to know. Because when I take it out of the incubator and I add reagent A and reagent B, the tube's going to turn red. It will turn red if there's any nitrite in the tube. So if my microbe has turned or reduced nitrate to nitrite, there'll be lots of nitrite in the tube, and the tube will turn red. Now, if it doesn't turn red, my next step is to put zinc in. There's really 2 possibilities if it's not red after I put reagent A in and reagent B in. There's really 2 possibilities, isn't there? Either there's still nitrate in the tube, right? The organism couldn't turn it to nitrite. Or the organism turned it to nitrite and then kept going. Turned it to nitrous oxide or turned it to nitrogen gas. If I put reagent A in and reagent B in, and I get no red color, all I know at that point is that there's no nitrite in the tube. So I need to do a little more work to figure out what is in the tube. So I'm going to add zinc, because if there's nitrate still in that tube, the zinc is going to reduce it to nitrite, and the tube is going to turn red. But that is a negative result because the microbe didn't do that reduction. The zinc did. The microbe didn't do anything. The microbe left the nitrate alone in the tube. When I added the zinc, I know there must have been nitrate in there because zinc can convert nitrate to nitrite. And once there's nitrate in there, the tube turns red. So if it's positive, if it's red after reagent A and reagent B, my microbe is positive for nitrate reduction. If it's red after zinc, my microbe is negative for nitrate reduction. But there is still another question. What if the reason there was no nitrite in that tube is because my microbe kept reducing it? What if my microbe made nitrous oxide or made nitrogen gas from it? And that's why there's no nitrite in there. I still need to do some more work. So if I add the zinc in and it still doesn't turn red, I've got two possibilities left. Either my microbe reduced the nitrate to nitrous oxide, or my microbe reduced the nitrate to nitrogen gas. How am I going to know? I'm going to put a Durham tube into this test. Remember what a Durham tube is? It's just a little upside down test tube. You can put this little tiny upside down glass test tube into a bigger test tube to catch gas. Nitrogen, N2, is a gas. So I can catch it in my Durham tube. So if I add reagent A and reagent B and nothing happens, and then I add zinc and nothing happens, the next thing I need to do is look in my Durham tube. If there's gas in there, it's nitrogen gas. If there's no gas in there, then the microbe turns the nitrate into nitrous oxide. Very, very complex test. Now, because I know that this is such a complex test, I made a couple of additional slides for you to help you with your notes. Remember, the first thing you have to do is you have to inoculate this nitrate reduction test medium. You have to put some cells in it. You have to incubate them for 48 hours. On this slide, it's going to explain how to move forward from there. So after 48 hours, I'm going to add reagents A and B to the tube. If my organism was able to take nitrate and reduce it, add electrons to it, to nitrite, I'm going to get a red color in the tube because in this test, the presence of nitrite in the tube, along with reagents A and B, is going to create a red color. So that's positive. That's a positive result. The microbe took the nitrate and reduced it to nitrite. And the nitrite turned red when I added A and B, or the tube turned red when I added A and B. Positive result. I can say that my organism can reduce nitrate. Now, if I put A and B in and there's no red color, there's still two possible explanations. Either the nitrate is still in the tube, because my organism cannot reduce nitrate, or the nitrate was reduced to another product. It was reduced first to nitrite, and then it was reduced to either nitrous oxide or nitrogen gas. Two possibilities still exist, so I have to be able to figure out which is which. The next step is to add the zinc. If there's still nitrate in that tube, in other words, explanation one, my microbe can't reduce nitrate, so there's still nitrate in the tube. If there's still nitrate in the tube, the zinc will reduce it to nitrite, and the tube will turn red. There's nitrite present now, so the red color develops. But that's a negative test result, because the microbe didn't make that nitrite, the zinc made the nitrite. It tells me, though, that there must have been nitrate in that tube, because zinc is only able to take nitrate to nitrite. Okay, so negative test if it turns red after zinc is added. Now, what if I put the zinc in and it doesn't turn red? Well, then I know I have a positive result, even though I don't have red color. I don't have red color because there's no nitrite in the tube. But the reason there's no nitrite in the tube is because it was further reduced. It was either taken all the way to nitrous oxide, or it was taken all the way to nitrogen gas through the addition of more and more and more electrons. Either way, though, the nitrate was reduced. So it's a positive test. How do I know which product is in there? I just look at the Durham tube. I look for gas in the Durham tube. If there's no gas, I know that the reduction process ended at nitrous oxide. If there is gas, I know that the reduction process produced nitrogen gas. Now, questions. Questions for this. Very complex test, right? Got to know how to do this one. Not an easy one. Got to know not only how to do it, what order to put all the chemicals in, but how to interpret the color changes. All right. All right, so let's do a little experiment. For today, our bugs, our bacteria are going to be Streptococcus pneumoniae, Staphylococcus epidermidis, our old friend E. coli, and Pseudomonas aeruginosa. The first two organisms are gram-positive cocci. We've seen Staphylococcus epidermidis before. Streptococcus pneumoniae is new for us. This is a Streptococcus organism that, when it's pathogenic, can cause a pneumonia, hence its name. E. coli and Pseudomonas are both gram-negative, and they're both rods. We've talked about E. coli and Pseudomonas before. E. coli can be perfectly harmless. It can be helpful to us, and certain strains are pathogenic. It lives in our intestine. Pseudomonas can live in us as well and not cause us harm. It can also live in the environment, though. And there are some strains that are pathogenic. And remember, Pseudomonas is one of those organisms that we call an opportunistic pathogen. Because while on a healthy person, it probably won't cause any problems. But when you have a patient who is very ill with some other problem, and their poor immune system is working very, very hard at that moment, Pseudomonas will take advantage of that. And Pseudomonas will become a pathogenic organism in that situation. In other words, it takes advantage of the opportunity to cause disease. Pseudomonas can cause disease in all kinds of immunocompromised patients. They don't have to be immune suppressed. They don't have to be an AIDS patient or a transplant patient. It's just a patient who is already really low. They're ill. They're ill with another problem. Their body is fighting to get better. And that little Pseudomonas is going to take advantage of that and cause disease. Pseudomonas is particularly problematic in burn patients, just like Proteus. It takes advantage. It takes advantage of an opportunity, basically, to cause disease. All right, so those are our 4 bugs. Here's our oxidase test. Remember, we're looking for purple color for positive, no color change for negative. Nitrate reduction test, well, this one's a little harder, right? It depends on where we are in this flow chart as to how we interpret it. I like this flow chart. It's another way to summarize this. Incubate for 48 hours, add reagent A and reagent B. This is showing us the actual names of these 2 chemicals. Don't worry about those names. You can call them reagent A and reagent B. We're going to add those in and look at the color. If it's red at that point, it's positive for nitrate reduction. If it's not red, if it's colorless at that point, I got to keep going. Now I'm going to add the zinc in. If it turns red, my organism is negative for nitrate reduction. If it stays colorless, I got to look in the drum tube. If there's gas, I've got a positive reaction. Nitrate reduction occurred all the way to nitrogen gas. If there's no gas in the Durham tube, I have a positive nitrate reduction, and the product is nitrous oxide. Big, long process for that test. This is what nitrate reduction test broth looks like. It's very beige, very boring. So this is what it looks like before you inoculate it. Remember, there's a little Durham tube in here upside down. I'm going to put my cells in. I'm going to cook it for 48 hours. This is that red color change that we're looking for when we add in the reagents in the right order. And here are the first of our results. Streptococcus pneumoniae, negative for the oxidase test. Staphylococcus epidermidis, negative. So both of our gram-positive cocci came up negative on oxidase. When we look at our gram-negatives, E. coli and Pseudomonas, one of them is negative and one is positive. So E. coli will give a negative oxidase test. Pseudomonas will give positive. Let's think about what we know about E. coli. E. coli is a facultative anaerobe. E. coli can switch and swap back and forth between respiration and fermentation, depending on the circumstances. It's got a negative oxidase test. We said that if it comes up positive for oxidase, it means that that microbe has cytochrome C oxidase in it, which means it has cytochrome C in it, which means it uses oxygen as its final electron acceptor. E. coli came up negative for oxidase. How can that be? How can that be? What do you think?
Is it because can it be both?
Well, if it's negative on oxidase, It doesn't have cytochrome C oxidase. It doesn't have cytochrome C. So how in the world could it do cellular respiration? What do you think? We know it's a facultative organism, so it can do respiration and fermentation. So how is it doing respiration? How is it using its electron transport chain without cytochrome C?
Does it use another cytochrome?
That's right. There are other cytochromes, right? There are other cytochromes. There's cytochrome A, there's cytochrome B, there's cytochrome D, right? It's confusing, but just because you're negative on oxidase does not mean you don't have any cytochromes. It just means you don't have cytochrome C. Okay? It's just a trait. It's a trait that an organism has. It's hard because sometimes students look at that test and they say oxidase test, oxidase. And they think, oh, oxygen, cellular respiration, aerobic cellular respiration. You can't do that. The oxidase test tests for cytochrome C oxidase. And there are other cytochromes. So that's why, that's how E. coli can come up negative on that test. But now we know something interesting about Pseudomonas, don't we? Pseudomonas uses oxygen as its final electron acceptor, and it uses cytochrome C and has cytochrome C oxidase. So that's, again, another interesting fact about Pseudomonas and something we could test for in the laboratory pretty easily if we thought we had a Pseudomonas organism. Okay. These are our nitrate reduction test tubes. This one is our Streptococcus pneumoniae. Here's the Staphylococcus epidermidis. Here's the E. coli. Here's the Pseudomonas. Now, there's some labels on here because, of course, this is a very complex test. So, for the middle two organisms, for Staphylococcus epidermidis and E. coli, we took the tube out of the incubator and we added in reagent A and reagent B. They're labeled as 1 and 2 here, but don't worry about that. It's just A and B. And both tubes turned red. Now, we also added A and B to this tube and to this tube, but they stayed colorless. So then we added zinc. And after we added the zinc, this tube turned red. This tube didn't. So now we have some interesting results. The two tubes that turned red after reagent A and reagent B were added, is that a positive or a negative? Took the tube out.
Positive.
Good, very good. Took the tube out of the incubator, added A and B, turned red. That tells us that there's nitrite in both of those tubes. And nitrite comes from nitrate through that reduction pathway. So those two organisms are nitrate reduction positive. They are both able to reduce nitrate to nitrite. Now, what about the Streptococcus? The strep stayed colorless after we put A and B in, but then it turned red when we put the zinc in.
That's a negative.
That's a negative. The zinc is what turned it red. So though that organism, Strep pneumoniae, has a negative result for nitrate reduction. It is not able to reduce nitrate. Can't do it. Doesn't have the enzymes. Okay. And then we have our friend Pseudomonas, who stayed colorless after A and B, after zinc. Is that a positive or a negative test?
That's positive.
That's positive. That's exactly right. There's no nitrate, there's no nitrite in that tube. That's why it's still colorless. It's just that Pseudomonas didn't stop at nitrite. Pseudomonas went on and kept reducing the nitrate, just kept going, kept adding electrons to it. It's able to do that. It has the right enzymes to do that. So what do we have to do? How do we know how far the Pseudomonas took it? What do we have to do next with that colorless tube?
We have to check whether there's gas or not.
That's exactly right. You got to look in the Durham tube. You got to look in the Durham tube. So it's pretty hard to see from where you are, but there's a bubble of gas right here in the top of this tube. You can see a white line right here. Everything below that is liquid in the Durham tube. There's some gas up here. So let me skip this for just a sec. So Staphylococcus epidermidis, E. coli, positive for nitrate reduction. They are both able to reduce nitrate to nitrite. Streptococcus pneumoniae, negative for nitrate reduction, unable to reduce nitrate. Pseudomonas aeruginosa, positive for nitrate reduction. Not only did it reduce the nitrate to nitrite, but then it reduced the nitrite to nitrous oxide, and then it reduced the nitrous oxide to nitrogen gas. Okay. I'm going to put one more slide up here just to confuse you a little bit more. No. One more thing, though. One more thing to note about this test. Let's say you take a nitrogen reduction test tube out of the incubator after 48 hours. Before you put any reagents in, you see that there's a gas bubble in the Durham tube. Can you interpret that? And the answer is yes, yes, you can. That's a positive test result. There is nitrogen gas in that Durham tube. So in some labs, I'm not recommending it, I'm not, but in some labs, if you pull a nitrate reduction test tube out of the incubator and you see a gas bubble in that Durham tube, you're already done. That organism is positive. It produced nitrogen gas by reducing nitrate all the way to nitrogen gas. It would be a really good idea, though, to go ahead and go through the steps and just confirm. Just confirm it. Add reagent A and reagent B. You should see no red color. Add zinc. You should see no red color. Now you've confirmed it.
I have a question.
Yeah.
So if you're presumably like results like there were two, the E. coli and the epidermidis.
Epidermidis, yep.
Yeah.
That's a staph.
S. epidermidis. Yeah. So those were both positive.
Yeah.
So you narrowed it down to those two with this test, but that requires further testing
Exactly right.
of the
I mean,
microbe.
this test, this particular biochemical test is not going to tell me the difference between those two, is it? It's not going to
No.
tell me the difference. But
Yeah, that was my question.
yeah,
But it does
a
narrow
lot
it
of our
down.
tests are like that, right? Which is why we usually run several different tests.
Multiple.
Yeah.
Yeah.
And it's
Understood.
also really helpful to know what organism you suspect it might be. For example, Staphylococcus organisms live on our skin. They can be found up in our noses. They can be found in the back of our throats. They are gram-positive cocci. They tend to be on our surface. Organisms like E. coli, those are intestinal organisms. Those are fecal or enteric organisms. So a nitrate reduction test is not going to tell me the difference between Staph epidermidis and E. coli. But if I know from the get-go that the sample that came into the lab was collected from a patient who had a terrible skin infection, the likelihood of that being E. coli, pretty low. I'm non-existent. So I already suspect I've got a staph. So this test might help me. It might help me confirm that I have a staph. It's not going to tell me which staph I have. I have to do multiple tests for that. But the other thing that's very helpful, in addition to running multiple tests, is to have some information about where did this sample come from? If it came from a human, a sick human, what were they sick with? What were their signs? That's going to narrow things down a little bit. If it came from the outside, where did it come from? Did it come from water? Did it come from soil? Did somebody find it in a drain in a hospital cafeteria? You know, where did it come from? That gives you information as well. And gosh, when you compare staph epidermidis and E. coli, a simple gram stain will help too, right? Because staph epidermidis is a gram-positive cocci, and E. coli is a gram-negative rod. That's why we almost always begin in the lab with gram staining. It's super easy, super inexpensive, it's quick. So pretty much the first test that gets done is a gram stain. Okay, that's going to give you information right there. So yeah, it's a pretty impossible thing to pick one test and get an identification on a microbe. You can't do it. But with the right combination of tests, you can absolutely identify them. Good question. All right. Any other questions or comments about it? All right, very good. All right, well, that's all I have for you tonight, so I'm going to let you go. Don't forget to take a look at start taking a look at the immunology lecture if you haven't already. Don't hesitate to contact me if you have any questions about anything that you're watching or reading or anything. And I will see you next time.
I have a quick
Okay.
question before you go, or
Yeah,
actually when
sure.
they go.
Okay. All right, everybody. Have a good evening. Is there anybody else I can help? I've still got 4 people in here. Or 5 people in here. Oh, I'm sorry, I'm the 5th. Who else? Who needs to speak with me? Is it Tiffany?
Well, me, Mary.
Oh, Mary. Okay. Tiffany, are you still here? I'm going to help you. And Sarah? Okay. Awesome.
Okay, so I know I'm obviously sick, but I'm stressing about the test. So can we get it done?
You can absolutely take the test if you feel okay to do it.
Well, my fever is gone, so well, there's that. The shortness of breath kicked in hardcore yesterday and kind of today, but I'm starting to have a bit more energy. So
Well,
I'm
good.
just kind of on mental status. I just don't want to get behind. And
I get it.
I really just need to keep busy because
Yeah.
I have asthma, so this is hitting me hard. I
I'm
can't
so
really
sorry.
walk that far. Yeah, I can't walk that far. So
Yeah.
just keeping with school is just kind of helped me get through it.
Okay. Okay. Well, that's all I need to hear. So what I'll do is I'll make sure you can submit everything whenever you're ready. Okay.
Okay, perfect. Thank
Don't
you.
feel pressured, but if you feel up to it and you think it's just a nice distraction to do it, go ahead and do it. Okay. And
Okay, thank you.
if you're not, if you don't feel up to it, just don't do it. Okay. Listen, if we
Okay.
get to the end of this month and you have not been able to finish all this work, I will just adjust it for you. I won't include certain things if you're not able to get to them. All right. We're going to get you
Okay.
through this. You have a terrific challenge in this. So don't worry. All right. That's number one. Don't worry.
Okay, thank you.
Even if you can't do another assignment, we're still going to get you through the course. Okay.
All right, I
All right.
appreciate it so,
Take good
so
care of
much.
yourself. Take good care of yourself. That's all you need to worry about now.
Okay, thank you.
All right. We'll talk to you later on. Okay.
Okay, bye.
All right. Bye-bye.